I'm impressed. Thanks for coming. I hope you're not super bored. There's one thing here. I was told by the organizing committee that next week, one of the organizers is going to give a tutorial on a very similar subject. So I was asked politely, but firmly, that I should probably talk about something else. I'm not going to give the name of the particular organizer, but he was going to.
Fake news.
So we are going to talk about fake news too.
And just talk about whatever you want.
No, there's a bigger story, but we'll talk about fake news too. So I decided to change the subject, and for background information, I actually cite the single paper co-authored by this particular organizer for some background, which just appeared in Nature Reviews Genetics, so you may enjoy that. And this paper is briefly mentioned, very briefly, there. So maybe this is your chance to learn about it. So this is on patient-specific driver gene prioritization in cancer. So as many of you know, cancer is slightly different from all the germline diseases covered throughout the week. We are now talking about somatic mutations collected over a lifetime. But these somatic mutations are mostly passenger alterations. They are neutral and provide no growth advantage to the tumor. And very few of them provide an evolutionary advantage and are positively selected. At least that's what the theory is. We call them driver alterations. More and more, we just call them cancer genes. And these driver alterations are diluted, which makes their identification pretty tough. There are some genes which are frequently altered in tumors. P53 is the quintessential example, but then their frequency dies out pretty fast. And there are those drivers that are really rare. And in fact, we believe that there are some private drivers. The latest estimates, I didn't go to the last PCOG meeting. How many of you were there in June? The previous PCOG meeting estimated the number of patients, cancer patients, frequent cancer patients, who do not have a single known driver gene altered between 20% to 25%. Maybe that number went down a little bit, but we are talking about a significant number of patients with no known alteration in the potential drivers, as we know. So how do we figure out these infrequent or private drivers? There are a number of statistical methods to identify potential drivers. And as some of the recent events have shown, statistical methods are not great in figuring out catastrophe. I'm not going to mention any particular event in November, but let's move on and try to figure out whether there are other ways to predict events, especially catastrophic events. And I'd like to highlight two sets of papers. In particular, HotNet and its followers. They're not aiming to figure out drivers, but they were interested in figuring out how information flows in a network, especially in a protein interaction network or a gene interaction network. And they were dealing with a diffusion approach. They introduced a network diffusion approach to model information flow. And I'd like to highlight one of the papers. These two inspired our work. DriverNet is by Sohrab Shah's lab, which aims to relate genomic alterations as potential drivers to alterations in target genes in the form of expression profile changes. So they start with the premise that these sequence alterations drive the expression profile changes, and we have to somehow link them. They were just looking at a protein interaction network, and for every potential alteration, sequence alteration, they were looking at the neighbors to see whether there's any correlation. So based on these two papers, we came up with HIT'nDRIVE. As we all know, The old is the new. So the first version of HIT'nDRIVE appeared 3 years ago. The new version is going to appear very soon in GR, which is a combinatorial method that integrates genome and transcriptome data to prioritize genomic alterations. And here comes the main idea. We don't have statistics, and if you're stats out, maybe just like me, through the week, you're looking for something different, and we are going to use maximum parsimony as our guiding principle. We'll say, let's try to figure out the minimum number of potential alterations that may explain all, and if not all, a good number of aberrant expression profiles per patient, and we'll look at large cohorts. So for that, we formulate the problem in a combinatorial way. So we are given a protein or gene interaction network. We know some of these genes have been altered from a sequence point of view, and we believe that a subset of them are the drivers. So how do you prioritize the drivers from the others? That is the problem. And we are going to formulate it as a facility location problem. So how many of you are familiar with the facility location problem in the very basic way? So this is a combinatorial optimization problem. It's a classical combinatorial optimization problem, which was first considered in 1800s. And it's concerned with optimal placement of facilities in a network. So suppose that this is my network. Suppose I'm going to place some coffee shops in a new city. Suppose that there are K coffee shops. I'd like to place them so that my customers will always be pretty near to a Starbucks shop whenever they want to have coffee. So it can be any coffee chain, by the way. Either you have a minimax formulation where the goal is to minimize the maximum distance from each client, or there's a min-sum formulation where the goal is to minimize the sum of distances. Any one of the formulations will do. And you may want to think of it as here's a hint. If you'd like to spread fake news, look at a social network. You may want to place the fake news in optimal locations so that it spreads out pretty fast, right? You can come up with all kinds of scenarios where this is useful. In our scenario, we don't have intelligent agents. We don't have customers trying to find the nearest Starbucks. Or in the hospital placement problem, we don't have intelligent ambulance drivers finding the best route to reach a patient. We have agents, which are biomolecules, which are not intelligent and cannot compute shortest paths. They just do a random walk, we assume, in this protein or gene interaction network. And to capture the uncertainty of biomolecular interactions, we will come up with a distance that is based on these random walks. The distance will be the hitting time. I don't know how many of you have heard about the hitting time. It's the expected length of a random walk in a network that originates in a node, in the source node, and ends up in the destination node. Right? So it provides a measure. It provides a distance, which is not symmetric, because the expected number of hops to go from A to B is not necessarily equal to the number of hops to go from B to A in a random walk. And you're talking about expectations now. And hitting time is interesting. It generalizes to multiple sources. So suppose that you have, in the hospital placement problem, you have multiple ambulances trying to serve a particular patient in an emergency situation. Rather than sending one ambulance, suppose that all hospitals send one ambulance independently. They're all trying to find the patient, but they're blind. They don't know where they're going. The moment they notice the patient, they pick up the patient. Right? So these multiple ambulances are searching the patient, and the expected length of the minimum number of hops from any one of these hospitals to the patient will be my, oops. okay, is my multi-hitting time. So based on this distance, I can define now a random walk facility location problem, which is a generalization of the facility location problem. And I described this problem to some of the top people in approximation algorithms, facility location, and so on. They had never heard about that. This is one of the rare instances, in my opinion, where a problem in bioinformatics inspires work in computer science and discrete math in general, although they don't have a solution, I have to say. And the random walk facility location problem can be described as follows. I'd like to come up with with case facilities such that the multi-facility, the expected multi-hitting time from these three facilities to any one of these domains is as small as possible. So how many of you do understand the definition? Somewhat. Any questions? Yeah.
Why a random walk?
Okay. So at a very intuitive level, we are dealing with biomolecules who don't know where they're going, right? They are just, there are some random interactions, some happen, some don't. Eventually, the information has to flow in a random fashion from the source to destination. And in fact, in all the for example, the flow-based methods also make the same assumption, but rather than looking at the distance, they're looking at the asymptotic distribution of random walks. Eventually, the flow formulations boil down to how walks behave in the asymptotics. So that is not new. What is new is this distance notion.
So it's not possible. It's now sound stochastic model.
It is a maximum parsimony. Based on this distance notion, it's just one distance notion. You calculate it in any way you want. Now I'm trying to figure out the set of facilities that minimize this distance. The distance is stochastic, but the problem is combinatorial. Yeah?
Do you assume that each direction is equally likely?
Yes.
Yeah.
We tried a number of possibilities. You can do it. There's no restriction, but the results that I'm going to present assume that all edges are equally likely. But you can easily update. I'd like to minimize the number of parameters as much as possible. We try to add weights based on high C data, a number of things. Okay, so here's the bad news. The random walk facility location problem is NP hard. And in order to overcome the difficulty in solving the random walk facility location problem, we introduced the following estimate of multi-hitting time. Multi-hitting time, I don't even know how to compute. Hitting time you can compute from every node to every source node to every destination in roughly quadratic time. But multi-hitting time we introduced in this paper, and we don't know how to compute. We only have simulations. I tried to work with people who are experts in random walks, and we failed. So what we did instead is introduce this Kirchhoff-inspired approximation, which works in certain situations. We say, okay, these random walks are like currents, and if two random walks originate in the same location, same source, it actually is quite accurate. If they appear in different loci, then it's questionable. But it seems to work reasonably well in practice. So this is probably the weakest point of the paper, although it makes the problem linear and approximable. So here are some work on single source hitting times. And this latest amalgamation is practical. It seems to work really well. So I will now define the influence of one node over another as the inverse hitting time. The more distant a destination node may be, the less influence the source node may have on it, right? If A is really far from B, its influence on B will be small. So the inverse hitting time will be the influence. And it turns out that network centrality is not necessarily a good measure of influence. It is a good measure of incoming influence. So a node like P53 is a good receiver of influence, but it's not necessarily a particularly good influencer. So this makes this measure actually pretty good for identifying rare drivers, because there'll be some drivers which are not central, but their influence over the rest of the network can be pretty high. Yeah?
How asymmetric can this measure get?
Hugely asymmetric. Just think of the star graph.
Okay. Yes.
So if you're at the periphery, your influence to the center is huge, but the center has no influence over any one of the peripheral nodes. Yeah?
The inverse hitting time to what?
Oh, okay. So in this plot, it's the median. We are looking at, for every x, you're looking at y. So you'd like to measure how likely you're influenced.
But every x meaning
every node.
Every subset or every subset?
Every node. Oh, I see. This is single hitting time, not multi-hitting time. That's a good question. We didn't explore that. Anyway, so let's reformulate the random walk facility location based on this linearization. So we are going to reduce it based on this estimate on the weighted multi-set cover problem. And we'll have an ILP formulation for it, integer linear program formulation for it. So our goal is, I'm going to change the problem a little bit. Rather than insisting that I pick up k drivers, I'll try to minimize the number of drivers, and I'd like to make sure that the expression-altered genes get sufficient influence from the sum, the collection of these drivers. So my goal is to make sure that every single expression-altered gene receives a total influence, which is the sum of the inverses of the hitting times, which is my estimate for the inverse of the multi-hitting time, is high enough so that I'm convinced that these potential drivers really are driving the expression up or down. And that suddenly reduces the problem to a set cover problem. I'd like to cover as many expression-altered genes as possible with the minimum number of these drivers. They form sets. The sets are labeled with potential drivers. Each potential driver is a set label. And I'd like to cover as many genes as possible. So this is the set cover version of the problem. I'm not going to go through every single detail of the formulation, but there's one parameter, alpha. I don't insist that I cover every single outlier. There's a proportion of these genes that I'd like to cover. Maybe 90% of them, 80% of them. The more I'd like to cover, obviously, the more drivers I have to pick. So here's the ILP. If you're interested, obviously, you can find and go through it. But let me just summarize what's going on. So I have these patients. I figured out which genes have been expression-wise altered for each patient. And I have these genes that have been sequence-wise altered. If this particular gene has been altered in this patient, I put an edge between this gene and that gene, all genes that have been expression-wise altered. Right? And these are weighted with inverse hitting times. Now I'd like to make sure that I pick up the smallest number of these genes such that the incoming influence, the incoming inverse hitting times, for a vast majority of these expression-altered genes is above a threshold. So that is the formulation. Obviously, what I'll figure out will be very dependent on the network that I use. And here are a bunch of networks that we tried. The string network, the reactome, HPRD, and we tried to figure out on a breast cancer data set how our drivers change with respect to, for example, gamma. This is the percentage of fraction of expression altered genes that have been covered. So, yeah, the bigger the data set is, here you can see the differences between these data sets with respect to number of nodes and number of edges. There will be higher or lower number of drivers, but these drivers, the genes themselves, overlap quite a bit. They really don't depend much on the nature of the network. In fact, as we'll see, even if we perturb the network, the drivers that we get are by and large the same. So there's something about the general structure of the network that seems to be pretty robust with respect to how the algorithm works.
Gamma is the alpha from previous slide.
No. Gamma is the proportion of genes that are covered, expression altered genes that are covered. Alpha is something different. Alpha is, I'm sorry, you're right. Alpha is the proportion of drivers. I screwed up. And gamma is the incoming influence. It's the minimum incoming influence that I need. You're absolutely right.
And you speak here now on implementation, but how would you solve the ILP?
Oh, ILP through, okay, I readily admit we use commercial solvers or Groby.
So this is some commercial solver to your ILP?
Exactly.
Okay, so not
It's Cplex.
implementation.
No, it's Cplex. For this particular case, Cplex seems to work well. And I encourage, for those of you who are not familiar with ILP, it's an excellent general way to formulate a combinatorial optimization problem. It's very, very robust, and there are good commercial solvers, free of charge for academic use, that you can use. So it's a great tool, great general tool to learn and use in your research. Okay, there are some results with respect to randomized input data. So if you just randomize the connections and try to predict drivers in this breast cancer data set, this is TCGA. Pretty much on a randomized network, the overlap with known drivers, the cosmic drivers, is flat as you increase gamma. So the more influence you need, the more drivers you need. And in comparison, HIT'nDRIVE, as you increase the number of drivers, their overlap with known drivers also increase. So this just shows that there is some information maintained in the network in comparison to a totally arbitrary network. This is completely arbitrary. Maybe I shouldn't get into every single detail, but for example, these are the number of drivers identified for each one of the 5 cancer types that we considered: glioblastoma, ovarian, prostate, breast cancer, and colorectal. We show the number of drivers versus the copy number burden and somatic mutation burden in these plots. We'll get back to those later. And here are the proportion of cancer gene census genes that were captured by HIT'nDRIVE on the 4 types of cancers. This doesn't include colorectal. And here, the green genes, each row is a patient. And you see that the green genes show the CGC genes, cancer gene census genes, predicted by HIT'nDRIVE. The red ones are the CGC genes that have been altered in that particular patient, but are not picked by HIT'nDRIVE. And the orange ones are the genes identified by HIT'nDRIVE, which are not in CGC, and the purple ones are not identified by HIT'nDRIVE as a potential driver, although they have been sequence-wise altered. You can see that the vast majority of the genes that HIT'nDRIVE identifies are CGC genes, but there are those rare or private potential drivers. And it's especially acute in prostate cancer because our understanding of, for example, breast cancer with respect to driver genes is pretty good, but prostate cancer is relatively poorly understood. There's one other study that we considered. This study considered roughly 265 anti-cancer drugs. And they tested them on 1,001 cell lines covering 30 different cancer types. It's a pretty impressive study. And we wanted to predict how well HIT'nDRIVE identifies the drugs that work in particular patients based on which genes they target. So if they target a gene that we think is a driver for that particular patient, we say yes. Otherwise, we say no. Even though a particular gene may be a cancer census gene, we may still say no because we don't use that information. So in principle, CGC genes should do much better because that's how these people figure out what drugs to use against what particular patient, what particular tumor sample. But as you can see, without that kind of prior information, based on just simple computational means, using expression and genome alterations, we can predict the drug efficacy as well, if not sometimes better than the CGC genes. So if you look at network properties of cancer driver genes that we identified, there are some hub genes that always appear, but HIT'nDRIVE also identifies some low-degree genes, which are not hubs, as potential drivers, as they have a lot of outgoing influence. Yeah, this coma is the same. So one question is whether we capture anything on top of DriverNet. So DriverNet, if you remember, looks at only influence of sequence altered genes on their neighbors in the network. And there are several important observations. DriverNet selects a large number of drivers. So these are the drivers. These are the outliers covered by these drivers. The number of drivers picked by DriverNet are in blue. It's huge in comparison to the reds, which are HIT'nDRIVE. On the other hand, what they were able to cover with respect to this notion of influence in the network is a much smaller proportion of what HIT'nDRIVE suggested drivers cover. And the proportion of, even though they figure out, they return a lot of potential drivers, the proportion of CGC genes that they discover is generally much smaller. So, okay, cancer gene census and the overlaps with CGC genes provide a notion of measure of success, but it's not a very exciting measure of success. We said, is there a way to come up with a quantitative measure of how well these drivers work, these potential drivers work? And we decided to do some phenotype classification. We said we are going to take these drivers, and since we are going to do phenotype classification in large data sets for which genome alterations are not always available, we decided to look at the potential modules, network modules, small subgraphs that are seeded by these drivers, potential drivers, whose aggregate expression seems to work well, seems to discriminate tumors from non-tumors in the training data set. So we identify some modules, one module per potential driver, and then we apply it to the test data. And we first want to figure out whether our classification accuracy compares well with respect to some random modules. And the random modules are important to look at, and it's not surprising that sometimes they do classification with 90% accuracy, because there's this training phase. We start with a random arbitrary node. We look at the module surrounding it that best correlates with the phenotype, and then we use that. So then we set up this classifier and compare against our classifier, which uses just the modules that are seeded by our drivers. So this particular classifier uses all possible kinds of drivers, and one module per driver which best associates with the phenotype. So in comparison, we are doing well. And there's the unsupervised version of some of these module identification methods. And we seem to do particularly well in this unsupervised approach. I'm not going to get into the details. And here are some comparison of the supervised, unsupervised versions of HIT'nDRIVE in capturing, in classifying tumor versus none versus DriverNet in 4 types of cancers. And finally, I'd like to emphasize that the phenotype classification accuracy of HIT'nDRIVE in comparison to CGC modules, CGC seeded modules, seems to be pretty well, especially in prostate cancer. As I said, prostate cancer is not that well understood with respect to drivers, and our classification accuracy is much better than what the CGC genes provide. There are many other tests that we perform. For example, unsupervised classification of breast cancer subtypes. There are, as you may know, 4 major subtypes in breast cancer. If you do just a classification, unsupervised classification based on expression profiles, you do pretty poorly. As you can see, each color denotes a particular subtype. And
where is the triple negative? Can you see the triple negative cluster?
Which one is triple negative?
The HER2 negative,
Yeah.
PR negative, but I didn't label all the color.
So,
PR, I didn't see.
There you go. Same goes with, I'm sorry, we have already gone through this. Yeah, if we compare the phenotype classification accuracy of HIT'nDRIVE versus just significantly differentially expressed genes, again, there's a significant difference with respect to accuracy, especially when you look at luminal A or luminal B. We can also reasonably well predict survival outcomes. I'm not going to get into that. But you can differentiate low risk versus high risk tumors. And finally, coming back to the drug efficacy study, we can reasonably well identify whether a particular drug, based on its target, would work in a particular patient or not. And this is just a plot on how well we do. In 80 to 90% of the drugs tested in the study, HIT'nDRIVE correctly predicts the drug efficacy in 80 to 90% of the samples. So it's pretty good in general. Not only it identifies the driver gene, by the way, but also it identifies the pathway reasonably well. You have to see some examples. I'm not going to get into that. In summary, HIT'nDRIVE is a combinatorial method to capture the collective effects of sequence aberrations on possibly distinct outlier genes based on what is called the random walk facility location problem. In this study, we introduced this multi-source hitting time notion, and we came up with estimates for that, applied it to large-scale networks. Hopefully, people will use it to contextualize multi-omics data sets in therapeutic decision-making. But I have to say one thing. I encourage, if there are those of you who are into social networks, any type of network analysis, to consider this formulation in cases where you're dealing with not very intelligent agents, where, say, you're talking about rumor spread, false news spread, how Bitcoin, for example, goes around in a social network, to see whether you can predict anything. I thank you and thank my collaborators and my funding agencies, and tell you that we are looking for postdocs on problems in cancer genomics, pharmacogenomics, proteogenomics, network biology, you name it, we do it. So thanks, and if you have further questions, please.